Okay, everybody, this is the back end of Chapter 24 on metabolism, nutrition, and body temperature. And what we're going to be looking at here is how the different macromolecules move through the pathways of anabolism and catabolism, what their fates are, and what their roles are. So we're going to start out talking about protein. Now, proteins will deteriorate over time with use, and so they need to be continuously broken down and recycled. But we don't just throw everything away when we recycle the protein. We break it down into its component parts. Remember that the component parts of proteins are the 20 amino acids that serve as the building blocks of every protein in the body, and that these amino acids can be grouped into different categories. They can either be charged, which means that they have an electrical charge at neutral pH. They can be nonpolar, which means that they tend to associate either with themselves or with components such as lipids. And they can be polar, which means that they prefer to associate with either charged or polar molecules or water. And by changing the sequence of the arrangement of these different building blocks and changing the length of the polymer that you build, you have an almost infinite variety of tools that the cell can use to perform metabolic tasks. Protein's not stored in the body as a fuel. It's used in the body as a tool. It's used in enzymes. It's used as membrane proteins, and it's used as structural proteins, such as the skeletal muscle proteins, myosin and actin, just as one example. When dietary protein is taken in excess, amino acids end up being oxidized for energy and converted to fat for storage. Now you might ask, well, how can this happen? Well, amino acids, by definition, have an amino group and an acid group on a carbon skeleton, and then they have a side chain that will vary in composition depending on the amino acid that we're referring to. When we oxidize amino acids, one of the things that we do is we remove the amino group and convert it into a keto group, and I'll illustrate how that works in just a minute. But the bottom line is that most of the amino acids that undergo deamination or transamination can be shuttled into the Krebs cycle, and as a result, the energy contained in those chemical bonds can be extracted and used to generate enough ATP to run the cell. In the event that you don't need them for energy, they can be also stored as fat because, again, these Krebs cycle intermediates have the capability of being converted into glycerol, which can then be condensed with fatty acids, and then this can be stored in our adipose tissue for later use. This is our energy storehouse between meals. So when we oxidize them, first we deaminate the amino acid, or we transaminate it, and then we convert it into either pyruvate or a keto acid intermediate in the Krebs cycle. Examples would be oxaloacetate or alpha-ketoglutarate. Any of these are possible intermediates that can feed directly into the Krebs cycle, just to name a few examples. The events include transamination, oxidative deamination, and keto acid modification. In transamination, what happens is that we steal the amino group off of an amino acid and we convert it to a keto acid. Now, an example would be glutamate. Okay, glutamate is an example of a negatively charged amino acid. If we strip the amino group off of glutamate, we produce alpha-ketoglutarate, which is a Krebs cycle intermediate. Glutamic acid can then be derived from this. This can also be converted into an amino acid by the addition of an amino group to the keto group and then be converted back into a keto acid. So these acids and these amino acids and these keto acids can be converted back and forth into each other. Oxidative deamination in the liver generally rids the body of toxic ammonia. This ammonia can either be excreted directly in the urine, or it can be used to generate urea which is a much less toxic nitrogenous waste that can also be disposed of in the urine. The ammonia removed from glutamic acid is used to generate now alpha-ketoglutarate. This removed ammonia is combined with carbon dioxide, and the urea that results is then excreted in the urine stream. We don't want it to build up to any high concentration in the blood or in the interstitial fluid, because one of the properties of urea is that it's a powerful protein denaturant, and as a result, as it builds up, it can cause proteins to lose their shape, and as proteins lose their shape, they lose their function. And so, as an example of what might happen if this were to occur, if these proteins lost their shape, they wouldn't be able to catalyze the chemical reaction that they help mediate, and that chemical reaction might stop. Okay, so that's an unacceptable result. And this is why the kidneys are designed to get rid of these nitrogenous wastes as soon as they're generated, so they don't end up with conditions like uremia and azotemia. Uremia meaning urine in the blood, azotemia meaning nitrogenous wastes in the blood, because this can lead to seizures, coma, and ultimately death. Now, if we look at keto acid modification, these acids can be formed by transamination and altered to metabolites that can feed directly into the Krebs cycle. An example is alanine and pyruvate. They can be converted into each other through transamination or deamination. We can also extract acetyl-CoA and alpha-ketoglutarate and oxaloacetic acid as some of these keto acid intermediates as well. Okay, acetyl-CoA feeds directly into the Krebs cycle. Alpha-ketoglutarate is a Krebs cycle intermediate itself, as is oxaloacetate. These reactions are reversible. Pyruvic acid can be converted into glucose in gluconeogenesis. Pyruvic acid can also be converted into alanine and used to synthesize protein. So we can go back and forth. And where a lot of these reactions occur is in the liver. And so what you're looking at here is just a model of the liver, and we're looking at the different steps that allow us to go from an amino acid to a keto acid and back again. So as an example here, an amino acid plus a keto acid, here the keto acid happens to be alpha-ketoglutarate, is converted now into a different keto acid and glutamic acid, which is an amino acid. Now this can be used to synthesize protein, or it can itself be deaminated. Okay, we can remove the amino group from it, and this ammonia can be reacted with carbon dioxide to form urea, which the kidneys will excrete. Okay, in oxidative deamination, the amine group of the glutamic acid is removed as ammonia combined with carbon dioxide and formed used to form urea. Now the reason this is called oxidative deamination is because when we strip the amino group off, we generate a keto group in its place, and that represents essentially the gaining of oxygen by that particular molecule, and that satisfies one of the definitions for oxidation. So the resulting molecule, that the keto acid that comes out of this, has less chemical potential energy than the amino acid that went in. Here's another example. Okay, the keto acid that's generated as the other product in this chemical reaction can be modified. Okay, this modified keto acid can be turned into one of the Krebs cycle intermediates, like oxaloacetate, for instance. These are formed during transamination. They can be easily isomerized so they can enter the Krebs cycle pathway, and then we can extract energy from them. So this is just showing you the critical role of the liver in doing this. So the bottom line is what? The bottom line is that the liver can convert amino acids from dietary protein into keto acids that can enter the Krebs cycle and serve as an energy source. At the same time, the liver can also take Krebs cycle intermediates, keto acids, and transaminate or aminate them and turn them into amino acids that can be used to synthesize protein. So we can go back and forth. So this is really where anabolic and catabolic pathways can intersect. Okay, amino acids are the most important anabolic nutrients. They form all proteins and they form the bulk of functional molecules in the body, and they are hormonally controlled. Okay, where we use them and how we use them is controlled by these hormone secretions in the bloodstream. They require a complete set of amino acids. Essential amino acids we have to get in the diet because the body does not have the ability to generate those de novo. We can synthesize between 225 to 450 kilograms of protein during our lifetime. Now, there's an interesting question to ask. Let's imagine for a minute that we didn't get some of the essential amino acids. What might the consequence be? Well, the consequence would be that we wouldn't be able to synthesize a large variety of proteins, and we would begin to see some of those effects. If you can't synthesize proteins, if you can't engage in anabolism, then cell function begins to break down. The result could be a deficiency in tissue function or even tissue death, and eventually a removal of homeostasis into the disease state, and then perhaps even resulting in death. So we need to have complete protein in order to survive. And how do we get that? Well, if we are an omnivore or carnivore, it's easy because we are what we eat. If we eat meat, meat is complete protein, and as a result will satisfy that dietary requirement. But if you're a vegetarian, you have to be very careful because different types of plant material contain different amino acid components, and you may not get all the necessary amino acids if you simply eat one type of vegetable. So you generally have to combine different types of vegetables in order to get complete protein. As an example, if you like to eat rice, you should augment those dishes with beans in order to get complete amino acids and be able to make complete protein. Otherwise, you're going to have deficiencies. Okay, catabolic anabolic steady state. This is a dynamic state in which organic molecules, with the exception of DNA, are continuously broken down and rebuilt. Organs have different fuel preferences, meaning that they like to use different fuel sources. So what they do is they derive their energy from nutrient pools. These are stores of amino acids, carbohydrates, and fats. There's three interconvertible pools of nutrients in the body. These are amino acids, carbs, and fats. The amount and the direction of conversion is directed by the liver, the adipose tissue, and the skeletal muscle. The body's total supply of free amino acids is called the amino acid pool. That's the definition. Proteins are often lost in the urine. They're lost by the hair and by shedding of skin and nails. They are a source for resynthesizing body proteins, for forming amino acid derivatives, and also for gluconeogenesis, the generation of new glucose from non-carbohydrate precursors. They're easily interconverted through key intermediates. These differ from the amino acid pool. Here we're talking about the carbs and the fats taken together. Fats and carbs are oxidized directly to produce energy. Amino acids have to first be converted into a carbohydrate or into a keto acid in order to extract energy from them. We can't directly use the amino acid right off the bat. Excess carbs and fat can be stored in the body, primarily as animal starch or as adipose tissue. Amino acids, however, are not stored as protein. The body doesn't set aside amino acids as a fuel store for future use. Most of our protein has a direct function. It either carries out an activity in the body as in structural proteins, enzymes, and membrane proteins, or it can serve, for instance, as cytoskeletal components. But we don't have vast amounts of protein that we set aside strictly to use as a fuel source. Now what you're looking at here is just the method by which we can interconvert proteins, carbs, and fats in order to either generate Krebs cycle intermediates that we can use directly for energy, or to generate amino acids that we can use to biosynthesize protein. And so you can see here, if we follow each of these flows, proteins can be broken down into amino acids. These can be transaminated into keto acids. The ammonia can be eliminated via urea through the urinary stream, and the keto acids can go directly into glycolysis and enter from there the Krebs cycle and be used for energy. By the same token, okay, we can rob glycolysis of intermediates, use these to generate keto acids, which can be then transformed into amino acids by transamination. Here you can see Krebs cycle intermediates. They can serve as a source of alpha keto acids that can be now transaminated into amino acids and used to synthesize proteins as well. If we look at fat, we have a slightly different arrangement here. Fats, remember, are a condensation of fatty acids and glycerol. Now, glycerol is easily converted into glycolysis intermediates as glyceraldehyde 3-phosphate, but the fate of the fatty acids themselves is completely different. The fatty acids can be converted in 2 carbon steps into acetyl-CoA, and the acetyl-CoA can directly enter the Krebs cycle. And remember, I drew for you guys a triglyceride in class just last Saturday, and I was trying to impress upon you the fact that there's a tremendous amount of chemical potential energy locked up in fatty acids because of the number of carbon-hydrogen bonds in the molecule. And so you can see that for every 2 carbon chain that we strip off a fatty acid, we can enter the Krebs cycle via acetyl-CoA and crank out now requisite amounts of NADH, FADH2, and ATP directly. Now, acetyl-CoA can sometimes form these abnormal structures known as ketone bodies. This is often seen, for instance, in diabetics who can't utilize the sugar in their bloodstream, and so they have to rely strictly on fat in order to get their metabolic energy. And the problem, of course, is that with excess amounts of acetyl-CoA from fatty acid breakdown, these ketone bodies will form. And these ketone bodies are kind of a metabolic dead end because you can't really make anything from a ketone body. You can split it back up into acetyl-CoA, but the ketone bodies themselves kind of have nowhere to go. And so in individuals that are experiencing the metabolic condition known as ketoacidosis, what you very often see are these ketone bodies being exhaled as volatile hydrocarbons, and this is why people that are in ketoacidosis often have breath that smells like nail polish remover, because acetone is essentially a ketone and is a cousin of a lot of these compounds. And this isn't a condition that you want to be in long term, because as the pH of the body fluids begins to fall, what happens is that proteins will denature and membrane potential will change to the point where cells won't be able to carry out chemical reactions, and the phenomena of membrane potential will be perturbed to the point where nerve transmission will cease and muscle contraction will stop, and essentially that means your death. Okay, on to the next slide. Okay, we want to talk a little bit about catabolism and anabolism. Remember that catabolism is the extraction of energy from the chemical bonds that are inside the food that you eat, and anabolism utilizes that energy in order to construct macromolecules in order to carry out life functions. The blood concentrations of energy sources are equalized between the absorptive state and the post-absorptive state. In the absorptive state, what we're talking about is the food as monomers. It's being absorbed across the lining of the small intestine into the bloodstream. And the post-absorptive state is what happens between meals. If you eat 3 square meals a day, the absorptive state persists for about 4 hours during and after each meal, and the absorption of nutrients is occurring during this time. In the post-absorptive state, we see this after the meals, okay, whenever they might occur. In a typical setup where you have, breakfast, lunch, and dinner, the post-absorptive state might be seen in late morning, late afternoon, all night, when the GI tract is empty and energy sources are supplied by the breakdown of reserves. Now, what are your reserves? They're your fat stores and your glycogen stores. And so what we have to consider now is, what parts of the body need what type of fuel. The liver has this amazing ability to generate virtually any fuel type from the fat and the glycogen stores. In the absorptive state, Anabolism, that is the building of macromolecules, exceeds the breakdown of macromolecules for energy. As a result, nutrients end up being stored. For carbohydrates, glucose makes up the major cellular energy fuel. It's the entry point for glycolysis. It's converted in the liver to glycogen or into fat. Synthesized fat and protein is released into the blood for storage by adipose tissue as very low density lipoproteins, or VLDL. You might also know these as your bad cholesterol. Okay, the reason we call it bad cholesterol is because if your levels of VLDLs and chylomicrons and LDLs are consistently high, that indicates that you're constantly eating more food than you're burning in terms of calories due to activity. So that's an unhealthy state of affairs, because that means an increase in body weight, and it also means an increase in blood triglycerides and cholesterol, which can lead to conditions such as arteriosclerosis, which leads to hypertension. Triglycerides are generated when lipoprotein lipase catalyzes lipids of chylomicrons in muscle and fat tissue. What lipase does is it cleaves the fatty acids from the glycerol backbone of the triglyceride. Most glycerol and fatty acids are converted to triglycerides for storage. So here we're talking about what's happening when we're trying to take the fuel that's being absorbed directly across the walls of the GI and then put it away for later use. Triglycerides are used by adipose tissue, liver and skeletal, and cardiac muscle as a primary energy source. The reason that fats are favored as an energy storage mode for the body as opposed to something like glycogen or protein is because fats are hydrophobic, and hydrophobic molecules tend to associate with themselves more than they associate with water. So hydrophobic molecules do not need to be solvated in order to be stored. In addition to that, if you look at the typical triglyceride, you have an abundance of carbon-hydrogen bonds, which means that there's a lot of chemical potential energy stored inside these molecules. And so the bottom line is that because of its nonpolar nature and because of its high amount of calories per gram, it's an excellent way to store energy for future use. We take up a minimum of volume and we store a maximum of calories. If we're looking at amino acids, excess amino acids are deaminated and used for ATP synthesis, or they're stored as fat in liver. Essentially what happens when these amino acids are deaminated is they're converted into keto acids, and these keto acids can serve as Krebs cycle intermediates, which means they can be used to generate ATP directly and also used to generate NADH and FADH2 in the Krebs cycle. In the event that they're not needed for ATP synthesis, they can be converted into fat, and the way that this happens is again by deaminating them and then converting them into keto acids, which can then be used to synthesize fatty acids. Most amino acids are used in protein synthesis, so they're primarily an anabolic component of the body. And so what you're looking at here is just some of the major events that take place in the absorptive state. Here we see the major metabolic thrust, which is anabolism and energy storage, as we absorb these monomers across the lining of the small intestine. Amino acids are used anabolically to generate protein. Glucose can be converted into glycogen, which is a glucose polymer, and we find that primarily again in liver and in muscle. But glucose can also be converted into fat. Glycerol and fatty acids can be converted into triglycerides, and those obviously can be stored in adipose tissue. Major energy fuels include glucose, which can immediately enter glycolysis and be used to generate ATP via glycolysis, the Krebs cycle, and the electron transport chain. In the liver, amino acids are deaminated and they're used for energy, or they're stored as fat. Okay, so the amino acids, when they are stripped of their amino group, are transformed into keto acids. These keto acids can be used to synthesize triglycerides, or they can be used directly in the Krebs cycle to generate ATP. Okay, and remember that a keto acid essentially, I'll draw one for you here. Okay, an amino acid and a keto acid. Let's just draw the general structure. Okay, so this would be an amino acid. Let's just draw an example of amino acid. This will be alanine. Okay, alanine is one of the nonpolar amino acids. This would be the carboxylic acid group here. Okay, the amino group would be over here. And the R group in the case of alanine is simply a methyl group. Remember that the R group on an amino acid can be any of 20 different side chains, and that amino acids are grouped as either nonpolar, polar, or charged at neutral pH. Okay, so there is alanine. Now when we convert alanine into a keto acid, what we do is we remove the amino group and we generate now an alpha keto acid. So the amino group becomes now a keto group. This is a keto group. Keto group is defined as a C double bound to an oxygen and on either side connected to a carbon. Okay, so this is an example of an alpha keto acid. Okay, and what do we bring in order to generate this? We can use oxygen. Okay, this is pyruvate, and remember that pyruvate is the end product of glycolysis, and it's also an entry point into the Krebs cycle. Forgive me here, my control's not the greatest with this pen. So that's pyruvate, and this is alanine, abbreviated ALA. Okay, and the only reason I wanted to show you this is to show you that you can convert an amino acid, okay, this is the acid group, this is the amino group, into a keto acid simply by deamination, or in some cases transamination. Sometimes we can steal this amino group and add it onto another keto acid. But the bottom line now is that this keto acid can serve as a source of fuel for the cell. In the same way, alpha keto acids can be converted into amino acids, which can be used for biosynthesis. Different enzymes in different tissues perform these events, go back and forth between either generating a catabolic intermediate or, in the case of an amino acid, an anabolic intermediate. So that's how it happens. Okay, so alanine here and pyruvate here. That's just one example of converting an amino acid into a keto acid. Okay, what you're looking at here is just the interplay between the small intestine and the other organs and the metabolically active tissues. Okay, so over here we have fat. Okay, and fat is found throughout the body. This is your liver, and this is your muscle. Okay, so let's take an easy trip here. The glucose component of your diet is absorbed into the bloodstream, carried to the muscle, where the muscle can store it as glycogen, and then in the absorptive state, this serves as a method of shoring up your quick energy. Okay. In addition to that, the amino acids that are derived from the protein that you eat can be carried by the blood to the muscle, where the muscle will use them to synthesize protein directly. If we continue this bifurcation, the amino acids we absorb dietarily can also be shipped over to the liver via the hepatic portal vein. They can be used to synthesize protein in the liver. The liver does a lot of that, blood plasma proteins being one example of many. They can also be converted into keto acids. The keto acids can be used directly to generate energy, okay, because they can be Krebs cycle intermediates. They can also be used to synthesize fatty acids, which when condensed with glycerol are used to make triglycerides, which are stored as fat. Okay. What else can we do? Well, if we follow the glucose pathway to the liver, okay, glucose can be shipped to the liver via the hepatic portal vein, where it can be stored as glycogen for later use as instant fuel, or it can be used to synthesize fatty acids, okay? And the way that this works is that we can actually now take the glucose, we can split it, okay? We can form pyruvate, and that pyruvate can then be used to form dihydroxyacetone phosphate, and that's a starting point for fatty acid synthesis. Over here, we can take the glucose, we can send it into glycolysis, and then the glyceraldehyde 3-phosphate can be used to synthesize glycerol, which is the backbone for triglycerides. The triglycerides then can be stored as fat for later use as fuel. Over here, glucose that's shunted to the liver can be used to synthesize fatty acids, can also be used to synthesize glycerol, and these can be used to generate triglycerides, which are stored in the fat. If we look at this branch of the bifurcation, triglycerides, which are absorbed dietarily, can be shipped directly to adipose tissue, where they can be used to condense with glycerol and form triglycerides, which are stored as fat, or they can be used to, they can be lysed, basically, they can be split to form fatty acids. The fatty acids can be used to form glycerides with glycerol as well. Okay, so basically fat can be broken up into its components and can be resynthesized into fat in the adipose tissue. And then finally, glucose can be used directly for energy in all tissues via glycolysis, the Krebs cycle, and the electron transport chain. The absorptive state is primarily controlled by insulin, which is secreted when we have basically high levels of blood glucose, is a signal to the metabolically active tissue to take the glucose out of the blood and use it for energy. So elevated glucose levels signal insulin secretion. Intestinal gastric inhibitory peptide and parasympathetic stimulation are also triggers for insulin secretion. And the bottom line is that as long as the insulin signal operates, you can utilize the glucose that's in the blood. It's when the insulin either is not made due to damage to the beta islet cells, such as in juvenile diabetes, or insensitivity to the insulin signal, as is seen in type 2 diabetes, that we have a problem. And the problem is that glucose levels in the blood continue to climb, and we end up actually spilling glucose in the urine in a condition known as glycosuria. In this event, what happens, and we're talking here about diabetes, is that the primary fuel is going to be fat. And you cannot completely combust fat without a significant number of carbohydrate intermediates. And so the byproduct of that incomplete fat combustion are ketone bodies. And those ketone bodies end up as volatile compounds that are expelled when you exhale. And this produces a condition known as ketoacidosis, because these compounds also have as their effect to lower the pH of body fluids. So this is not a good long-term condition for the body to be in. Insulin is a hypoglycemic hormone, and it enhances facilitated diffusion of glucose into muscle and fat. Brain and liver take up glucose without the need for the insulin signal. Glucose oxidation is required for the production of ATP. It's also required for glycogen and triglyceride formation, the active transport of amino acids into tissue cells, and protein synthesis, and it's also necessary as a signal. It inhibits glucose release from the liver, and it also spurs gluconeogenesis. Basically, what insulin's designed to do is to try and reduce blood glucose levels in any of these particular pathways. Gluconeogenesis, remember, literally means the making of new sugar. Neo, new, genesis, to make, glucose, sugar. And so what happens here is that we inhibit this, okay, because we don't need it, right? The glucose is already in the blood, and so what we're in is a storage mode or an energy burning mode in the post-absorptive state, and so we inhibit these two liver functions. So again, insulin helps facilitate a diffusion of glucose into muscle and fat. This opens the doorway to use it for energy. It also promotes glycogen and triglyceride formation so that we store it as a temporary fuel in our body's pantry. Promotes active transport of amino acids into tissues. Promotes protein synthesis, so it has an anabolic effect. And it inhibits anything that's going to try and elevate blood glucose levels because they're already elevated. And this is just a peek at how it is that insulin directs nearly all the events of the absorptive state. So here we see the absorptive state. In the absorptive state, what is happening? Well, we're absorbing nutrients across the wall of the small intestine. Among those nutrients is glucose. The elevated blood glucose stimulates the beta cells to crank out insulin from the pancreas. Okay, this is the endocrine pancreatic function. This elevates blood insulin, targets tissue cells, increases active transport of amino acid into tissue, increases protein synthesis. It also increases facilitated diffusion of glucose into tissue cells for use as energy, to synthesize fatty acids and glycerol, or to store glycogen. Okay, this is an energy yielding step. These are energy consuming steps. So what happens in diabetes mellitus? What we have is a dysregulation of the insulin signal. Inadequate insulin production or abnormal insulin receptors lead to being being unavailable for most body cells. Basically, the signal isn't working, and so the glucose remains in the blood. Glucose is, as a result, lost in the urine. This also increases the volume of urine, so this is a condition called polyuria. So in people with diabetes mellitus, you tend to see polyuria and glycosuria. It also puts a huge strain on the kidneys because of the difficult time that the proximal convoluted tubule has trying to reload all that glucose back into the bloodstream. That's why it ends up in the urine. Fats and proteins, as a result, are used for energy, and this results in the generation of ketone bodies, producing metabolic acidosis, protein wasting, and weight loss. But this cannot persist for very long because of the fact that we are in metabolic acidosis. If this persists for too long, then we're going to have a severe effect on membrane potential, muscle contraction, and nerve transmission. In the post-absorptive state, we see breakdown of fat, glycogen, and protein, and this exceeds the anabolic reactions, basically the synthesis of macromolecules. So we have net synthesis of fat, glycogen, and protein ending. The goal is to maintain blood glucose levels between meals. This makes glucose available to the blood, and it promotes the use of fats for energy, so it has a glucose-sparing effect. It saves it for organs that need it the most, and those include things like the brain. What are some sources of blood glucose? Well, you've got the breakdown of glycogen in the liver. This is termed glycogenolysis. We also see this in skeletal muscle. We see the breakdown of fat in adipose tissues and in the liver. That's called lipolysis. The glycerol is what's used for gluconeogenesis in the liver because the glycerol can be converted into glyceraldehyde 3-phosphate, which is again a, glycolytic intermediate. This can be isomerized into dihydroxyacetone phosphate, and then that is part of the biosynthetic pathway that eventually leads to glucose biosynthesis. It also can come from the breakdown of cellular protein. This is a major source that happens during prolonged fasting. Again, what's the idea here? The idea is that the amino acids can be deaminated or transaminated to form Krebs cycle intermediates. Those Krebs cycle intermediates can then be used to synthesize pyruvate, which can be used to synthesize dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and then that's the biosynthetic pathway that ultimately leads to glucose. The amount of fat in the body determines how long we can survive without food. Basically, the more fat you've stored, the longer you're going to be able to live without eating. And so here's what's happening in the post-absorptive state. In the post-absorptive state, nothing is crossing the small intestine because we're not eating. So where does our fuel come from? Our fuel stores. Okay. Proteins can be broken down into amino acids to be used as fuel. Glycogen can be broken down into glucose to be used as fuel, and triglycerides can be split into glycerol and fatty acids to be used as fuel as well. Major energy fuels include glucose and fatty acids and ketones, and this produces large amounts of ATP available for cellular work. Glucose is provided by glycogenolysis and gluconeogenesis. Fatty acids and ketones are provided by your fat stores. What happens in the liver? Amino acids get converted into glucose by converting amino acids into keto acids, which can then be used to synthesize glucose in, again, the act of gluconeogenesis. And then what's the glucose used for? It's used for energy. So this is just showing you some of the different pathways that lead ultimately to an adequate supply of nutrients between meals using the appropriate fuel source. Okay? The nervous system, for instance, primarily feeds off of glucose. It really doesn't understand any other fuel. Your fat can provide now fatty acids and glycerol. The ketone bodies can also be produced as a result of fat breakdown, and ultimately this is used in most tissues to generate large amounts of energy. Over here we can see the stored glycogen that is built up inside the liver through glyconeogenesis. It can be broken down into glucose. Okay, and where does the glucose go? The glucose can go into the blood, where it will be a direct fuel for organs that can understand glucose as a fuel, that being primarily the central nervous system. Post-absorptive state, what we're doing, again, breaking down components in order to meet our fuel needs. Protein in muscle can be broken down into amino acids, which can be converted to keto acids and used to synthesize glucose. Glycogen can be converted into pyruvate and lactate. This can be used to generate glucose as well. Fat, primarily in the form of triglycerides, can be broken into glycerol and fatty acids. The glycerol and fatty acids, the glycerol can be used to synthesize glucose, and the fatty acids can be used to generate keto bodies, which can be used to generate fuel, okay, and also can be used by metabolically active tissue to generate fuel, not only in the liver, but elsewhere in the body. So that's basically what's going on. We're breaking down our protein, we're breaking down our fat, and we're breaking down our glycogen, and we're sending the appropriate fuel to the appropriate organ in order to keep you alive. Because, again, we're not eating. Sympathetic nervous system inputs interact with hormones to control events of the post-absorptive state. This state is triggered by reduced insulin release as blood glucose levels drop. Moderate intake of beer, wine, and gin before or during a meal results in the improved use of insulin. This lowers blood glucose, but no increase in insulin is seen. Glucagon is a hormone that acts counter to insulin. It's a hyperglycemic hormone, and its release is stimulated by declining blood glucose and rising amino acid levels. So what's glucagon going to do? It's going to do anything it can to elevate blood glucose levels. So this includes gluconeogenesis in the liver and glycogenolysis in the liver, among many other effects. So we've already detailed those. It also triggers lipolysis in adipose tissue, which causes fatty acid and glycerol to enter the blood. It also modulates the glucose effects after a high-protein, low-carbohydrate meal. And so here you can see some of the effects of glucagon. Okay, we see a decline in plasma glucose. The alpha cells release the glucagon. What happens is that the blood levels of the hormone rise. This stimulates glycogenolysis and gluconeogenesis in the liver. This elevates plasma glucose. And this has a negative feedback effect on the pancreas. Okay, so there's the negative feedback loop. What else does it do? It stimulates fat breakdown. It increases plasma fatty acids, and this fat is used by tissue cells and has a glucose-sparing effect. And now we're able to meet our energy needs between meals. Okay, so again, to review, insulin will reduce blood glucose levels and promote the storage of fuel. Glucagon will elevate blood glucose levels in order to meet fuel demands between meals. So what are some of the hormonal and neural controls that regulate the post-absorptive state? Well, rising blood amino acid levels can trigger insulin and glucagon released. It's not a problem if we eat a balanced meal. If we have a high-protein, low-carb meal, we increase the stimulus for insulin release. If this isn't counterbalanced, this can lead to hypoglycemia, and this can result in brain damage. It's counterbalanced by glucagon release, which stabilizes blood glucose levels. Adipose tissue is innervated by the sympathetic nervous system. It quickly supplies glucose if the blood levels are low. Low plasma glucose is the result of the fight or flight response initially. So what we have to do is we have to elevate blood glucose levels as quickly as possible to meet the energy needs of this emergency situation. Low plasma glucose in the fight or flight response or for exercise results in fat mobilization and glycogenolysis as a result, again, of sympathetic nervous stimulation. As a result, blood glucose levels begin to rise and we're able to meet our emergency energy needs. This is initiated by sympathetic nervous system and epinephrine that comes from the adrenal medulla. Remember that the adrenal medulla is basically a modified postganglionic sympathetic neuron that instead of innervating a target, releases epinephrine directly into the bloodstream, and as a result you have these widespread effects. Essentially, the bottom line is that your blood glucose levels will begin to climb and your fuel reserves will begin to shrink. Metabolism and nutrient flow is also influenced by growth hormone, thyroxine, sex hormones, and corticosteroids. Hepatocytes are liver cells. They have thousands of metabolic functions. They process nearly every class of nutrient. They play a major role in regulating plasma cholesterol. They also store vitamins and minerals, and they metabolize alcohol, drugs, hormones, and bilirubin. In fact, your liver does so much for you that you couldn't live without it. In fact, the name liver comes from the ability to live. If your liver were to die, you would not be able to remain alive. Fortunately for us, the liver is one of the few organs in the body that has a property known as immune privilege. So in the event that you need a liver, you do not have to have a histocompatible donor. All you need is the proper blood type. The liver also has the ability, within reason, to regenerate in the face of damage, and that regeneration produces functioning hepatocytes. Let's talk a little bit about cholesterol. It's the structural basis of bile salts, steroid hormones, and vitamin D, and it's a major component of plasma membranes. It basically controls membrane fluidity. The more cholesterol you have in a plasma membrane, the less fluid the membrane becomes, and the less cholesterol you have, the more fluid the membrane becomes. Basically, when we mean fluid, we mean that the phospholipids can move in a two-dimensional arrangement around the cell. 15% of blood cholesterol is ingested. The rest is made in the body primarily by the liver, and it's lost from the body when it's broken down or when it's secreted in bile salts. Remember that bile salts are important in emulsifying fat so that lipase from the pancreas can effectively break the dietary fat down into fatty acids and glycerol, which can then be absorbed eventually as resynthesized triglycerides by the lacteals that lie just underneath the lining of the small intestine. It's also part of the hedgehog signaling molecule that directs embryonic development. Now that's something that may be a little arcane to you. Basically, what these molecules are a group of proteins that are called transcription factors. And what they do is they control the expression of particular genes that direct development. They do so by controlling processes such as organogenesis and morphogenetic movement. So without this signaling molecule, we would have problems making cells behave the way that they're supposed to during normal human development in utero. Lipoproteins transport water-insoluble cholesterol and triglycerides in the blood. They regulate lipid entry and exit at target cells, and they contain triglycerides, phospholipids, cholesterol, and protein. A higher percentage of lipids that are low density means that you're not in good health. Okay, so we don't want to have high amounts of what we call the bad cholesterol, okay, which are the VLDLs, the LDLs, and the chylomicrons. What we want to have is a lot of the HDLs. That's defined as the good cholesterol, because what that means is that you are in a fat breakdown mode as opposed to a fat depositing mode in the body. The reason we want to keep cholesterol levels low in the blood is because we don't want them to deposit on the walls of arteries and produce arterial sclerosis, which leads to hypertension. Lipoproteins are basically proteins that transport components around the body. Okay? Lipoproteins are conglomerations of lipids and protein, and they are the vehicle that move fat. The reason that you need the protein as a vehicle is because proteins are more water soluble than fat is, and so this provides a method of solubilizing the fat and moving it from and to the liver and from and to the skeletal muscle and delivering it wherever it needs to be delivered in order to use it for either energy generation or biosynthesis. HDLs are your good cholesterol. These are your high-density lipoprotein, and they have the highest protein content. LDLs are your low-density lipoproteins, and they are cholesterol-rich. And VLDLs are very low-density lipoproteins and are made mostly of triglycerides, while chylomicrons are the lowest density lipoproteins, and they are made almost completely of triglycerides. And here you can see the different components of the different types of cholesterol. Okay, so there's the chylomicrons, the VLDLs, and the LDLs. These are all what we consider to be quote-unquote bad cholesterol. Now they're important in the sense that they're part of moving fat around the body, but if you have a preponderance of these three classes of lipoproteins in your blood, that means that you're primarily in a fat depositing mode most of the time, as opposed to being a fat burning mode. You can see the different proportions of the different classes of molecules in each of these types of lipoproteins. The triglycerides here in yellow, the phospholipids in gray, the cholesterol in peach, and the protein in purple. The HDLs are your good cholesterol, okay? And what happens is that they return to the liver, and they represent now the breakdown of cholesterol and the elimination of cholesterol from the body. VLDLs transport triglycerides from the liver to the peripheral tissues, primarily adipose tissue. LDLs transport cholesterol to peripheral tissues for membranes, for storage, or for hormone synthesis. And HDLs transport excess cholesterol from peripheral tissue to the liver to be broken down and secreted into the bile. And they also provide cholesterol to steroid-producing organs. So you can see why an abundance of chylomicrons, VLDLs, and LDLs is indicative of a fat storage state in the body as opposed to a fat breakdown state in the body. Your total cholesterol should be 200 milligrams per deciliter or less. Levels greater than 200 milligrams per deciliter are linked to arteriosclerosis. The form in which the cholesterol is transported in the blood is important to measure. High HDL is thought to protect against heart disease. Greater than 60% or greater than 60 milligrams per deciliter is considered to be good. Less than 40% is not good. High LDLs lead to cholesterol deposits in the vessels. 100 or less are good is good. 130 or above is not good. Okay, and the same goes for the VLDLs and the chylomicrons. They, again, if they predominate, you're going to be depositing cholesterol in the vessels, leading to arteriosclerosis, leading to hypertension. The liver makes cholesterol. At a basal level, regardless of dietary cholesterol intake, is constantly synthesizing this complex molecule. It usually does so in response to saturated fatty acid levels. The ratio of saturated to unsaturated fatty acid affects blood cholesterol. If we look at saturated fatty acids, these stimulate the liver synthesis of cholesterol and they inhibit cholesterol excretion from the body. Unsaturated fatty acids enhance excretion of cholesterol and enhance cholesterol breakdown into bile salts. And so this is why you've probably seen a lot of advertising regarding healthy or unhealthy fat. Basically, polyunsaturates are fatty acids that have more than one double bond in the hydrocarbon tail. They represent a lower energy content than saturated fatty acids, which are all single bonds. The saturated fatty acids, by their nature, are going to have more carbon-hydrogen bonds, which means they're going to have a higher calorie content, which ultimately means that they're going to represent an energy imbalance. You're going to tend to store that as fat. Whereas the unsaturated fatty acids, by their very nature, have a lower calorie content, and they also, again, trigger these beneficial physiological activities. Trans fats are basically oils that have been forced to be solids. Examples include Crisco and margarine. They have a worse effect on cholesterol levels than saturated fats do because they increase LDLs and reduce HDLs. Unsaturated omega-3 fatty acids, which are found in fish, lower the proportions of saturated fat and cholesterol. They make platelets less sticky and prevent spontaneous clotting, which can lead to embolus formation, which can lead to heart attack and stroke. They also have antiarrhythmic effects on the heart, and they also lower blood pressure. So this is why it's recommended that we keep a lot of omega-3 fatty acids in the diet. You can also get them from nuts as well as from fish. and there are certain vegetables that are high in omega-3s as well. Other factors that affect cholesterol levels include stress, cigarette smoking, and coffee, which lower your HDL levels. Aerobic exercise and estrogen increase HDL levels and decrease LDLs. Your body shape is also determined by your cholesterol proportions. If we have an apple shape, which means a broad midsection, the fat is carried on the upper body is correlated with high cholesterol and LDL levels. If we have a pear shape, the fat's carried on the hips and the thighs, and this correlates with lower cholesterol and LDL levels. So again, your body shape can determine your cholesterol profile. What are some indicators for cholesterol level treatment? If we look at LDL levels in the blood, we can use this as an assessment of cardiovascular disease risk. It was previously used high cholesterol and LDL HDL levels as a measure of this overall lipid profile, but now it's found to be less reliable than other types of measure. Statins are cholesterol-lowering drugs. It's estimated that greater than 10 million Americans take statins. Okay, where does the energy come from in the food we eat? Well, it comes from the chemical bonds that are contained in those molecules. And the money bond in biological systems is the carbon-hydrogen bond. So essentially, the more carbon-hydrogen bonds you have in a particular molecule, the higher the calorie content is going to be. So the bond energy is released from food, and this bond energy is used to synthesize other molecules in anabolic activity. It also generates heat, so it's a form of body heat, and it's also used to power cell activity. So bond energy released from food has to equal your total energy output. Your energy intake equals the energy that's liberated during food oxidation, and your energy output can be immediately lost as heat. This is about 60%. It can be used to do work, and this is driven again by ATP. That's biological gas in the tank. And it can also be stored either as fat or as glycogen. And so that's how we shore up the body's pantry so that we have energy to utilize between meals. Nearly all energy from food is eventually converted into heat, and this heat can't be used to do work. It radiates off into the universe and contributes to the increased entropy of all of existence. That's the second law of thermodynamics. Heat energy warms tissues in the blood. It maintains homeostatic temperature, and it allows metabolic reactions to occur efficiently. If energy intake equals energy output, your weight is going to remain stable, but if they're not equal, you're either going to gain or lose weight. So let's talk a little bit about obesity. Your body mass index is equal to your weight times 705 divided by your height in inches squared. It's considered overweight if your BMI is 25 to 30. You're considered obese if your BMI is greater than 30. Higher incidence of arteriosclerosis, type 2 diabetes, mellitus, hypertension, heart disease, and osteoarthritis are conditions that accompany this particular condition. More adults and children are overweight than 20 years ago. Now, we have to be careful about this measurement, body mass index, because it's simply the weight of your body divided by your height, right? And what we have to think about here, okay, the weight in this body mass index is considered to be bad weight if it's adipose tissue. And so there's a simplifying assumption here, which is the reason that you have the weight that you've got is primarily due to the accumulation of what we call bad weight, adipose tissue. This breaks down, this BMI calculation breaks down if your excess weight is due to things like skeletal muscle. So if you're a weightlifter, you're going to have a very high body mass index, and that isn't going to necessarily be correlated with these negative health effects. So one of the ways that we get around this is that we use other ways to measure body fat. One of the ways we do this is we can weigh you in air and weigh you in water, and then we can compute the differential. The reason for that is that fat floats. So if your excess weight is primarily due to adipose tissue, you're going to weigh considerably less in water than you would in the air. Whereas if your excess weight is due to muscle tissue, muscle is much denser than fat, and the differential between your weight in water and your weight in air is going to be much less. Okay, this is one method that we can use to try and compute the percentage of fat in your body. The other thing we can do, we can use caliper measurements. We can use electrical impedance. There's all kinds of methods to compute this. So BMI is a very simple-minded calculation. It definitely has drawbacks. Okay, how do we regulate food intake? The arcuate nucleus in the brain and the lateral hypothalamic area, as well as the ventromedial nucleus of the hypothalamus, all have a role. They release peptides that influence feeding behavior. Some ARC neurons produce a compound called neuropeptide Y. This is a short-chain protein. And agouti-related peptides can enhance appetite. Other ARC neurons can produce pro-opiomelanocortin compounds, and cocaine and amphetamine-regulated transcript compounds can suppress the appetite. The problem with a lot of these, okay, these unusual small molecules, is that most of this work has been done in animal models. And the reason that animals eat is because they're hungry. And humans eat for other reasons. Okay, humans eat when they're hungry, but they also eat in differentiating emotional states. So a lot of this work sort of falls apart when we try to translate what we find in animal models to what we actually see in human beings. When we try to base drugs on these compounds, sometimes they don't necessarily work. Feeding behavior and hunger are triggered by neural signals from the digestive tract and blood-borne signals related to body energy stores. Hormones also have a role, and to a lesser extent, body temperature and psychological factors. These operate through brain thermoreceptors, chemoreceptors, and others, and food intake can be controlled either short or long term as a result of these controls. So let's look at some of the neural signals that come from the GI. High protein content of a meal can increase and prolong afferent vagal signals, and distension can send signals along the vagus nerve that suppress the hunger center. So this is our satiety reflex. The idea that we eat until we feel full and then we stop. Nutrient signals that are related to energy stores include increased nutrient levels in the blood that can suppress eating. These are related to rising blood glucose levels and elevated blood amino acid levels, as well as the blood levels of fatty acids. Hormones also play a role. Gut hormones, such as insulin and cholecystokinin, suppress hunger. Glucagon and epinephrine stimulate hunger. Ghrelin from the stomach stimulates appetite. Levels often peak prior to mealtime. But again, I have to point out, all of these findings have their basis in animal models. Okay, and remember that animals eat because they're hungry, completely only because they're hungry. Humans eat for other reasons, and so sometimes the cerebral cortex can override all this stuff. Leptin is a hormone that's secreted by fat cells in response to an increase in body mass, and it's an indicator of total energy stores in the fat tissue. It's supposed to protect against weight loss in times of nutritional deprivation. It's thought that a problem with leptin signaling may be one of the links to human obesity. Basically, we end up not responding to the leptin signal. We continue to eat. As a result, we gain weight. So we become insensitive to this particular hormone. Where does it act? Well, it acts on ARC neurons in the hypothalamus, and it suppresses the secretion of neuropeptide Y, which is an appetite stimulant. It also stimulates the expression of appetite suppressants, such as the CART peptides. Rising leptin can lead to some weight loss, but it is not a magic bullet for obese patients. High leptin levels in obese patients indicate that they're resistant to its action. The function is to lead to weight loss prevention during nutritional deficiency. And here we can see the interaction of these short and long-term controls and where they actually trigger hypothalamic responses. Okay, so you've got here the GI, okay, you've got stretching of the stomach, absorption of monomers from the breakdown of food, insulin and cholecystokinin, and then these other hormones, ghrelin, glucagon, and epinephrine. And then you can see where they act, okay? The distension acts on the vagal afferents, the monomers act as nutrient signals, and the insulin CCK are basically gut hormones, and these other components can act directly on the hypothalamus. Here, these three components act on the solitary nucleus in the brainstem, which has a direct effect via long-term controls on this class of neurons in the hypothalamus. Over here, pancreatic insulin and leptin from fat act on this class of neurons in the hypothalamus and this class of neurons, okay, the neuropeptide Y secreting neurons. What happens in the hypothalamus is that we have melanocortins that act now on these VMN neurons that and basically trigger the satiety sensation. Satiety is the sense of feeling full. The neuropeptide Y and AGRP secreting neurons target the LHA orexin releasing neurons, and this is an appetite enhancer. Okay, so these orexins basically make you hungry. And again, a lot of this comes from animal models, so take it with a grain of salt. Okay, additional factors that regulate food intake. Cold temperatures activate hunger. Stress can either increase the amount of food you want to eat, or it can decrease the amount of food you want to eat. And this is where higher brain centers come into play. Psychological factors, this is again higher brain centers overriding a lot of these hypothalamic interactions between the gut and the brain. Adenovirus infections can have a role, as can sleep deprivation and the composition of your gut bacteria. Your metabolic rate is measured by the total heat that's produced by the chemical reactions and the mechanical work of your body. It can be measured directly through a calorimeter, which measures the heat liberated into a water chamber, or indirectly through a respirometer, which measures oxygen consumption, which is directly proportional to heat production. It reflects energy body needs to perform most of its essential activities, and it's measured in the post-absorptive state, in the reclining position, in a relaxed mental frame of mind, and both mentally and physically, and at a normal room temperature, about 25 degrees centigrade. It's recorded as kilocalories per square meter of body surface area per hour. An example would be 70 kilogram adult with a BMR of about 66 kcals per hour, just as a typical measurement. It's influenced by body surface area, age, gender, body temperature, stress, and thyroxine. And one other very important factor, genetics. Okay? So a lot of your basal metabolic rate is born into you at fertilization. And the reason for that is that all of the proteins that carry out these various body functions have variations in their structure, just as every gene product has alleles. And as a result, some people are born with a quote-unquote high metabolism. Some people are born with a quote-unquote low metabolism. We can, through exercise and diet, control our body mass to a certain extent, but our genetics also have a large role to play. So the way I like to look at it is that At birth, you're born with an upper and lower limit on a particular trait. Here the trait would be body mass, okay? And it's your interaction with your environment that's going to control where within that window you lie. And so if you cut down on the calories and you exercise a lot, you're going to be towards the lower end of your biologically set window. Whereas if you eat a lot and don't exercise, you're going to tend towards the upper end of your biologically set window. That's an easy way to kind of wrap your brain around it. Your BMR increases as the ratio of body surface area to volume increases. It declines with age. Males have disproportionately higher basal metabolic rates primarily due to testosterone levels. It also increases with temperature and stress. Thyroxine increases oxygen consumption, cellular respiration, and basal metabolic rate, and physical training generally has little effect on basal metabolic rate. Although, again, the more muscle tissue you accumulate as a percentage of your body mass, the higher your basal metabolic rate is going to go, because muscle has a higher basal metabolic rate than fat does. So there's going to be an effect as a result of exercise. There's also going to be an effect because you're going to elevate your body temperature as you exercise. And it's also been shown that exercise increases, to some extent, the production of steroid hormones. Okay, so when we say physical training has little effect, maybe not in a direct sense, but maybe in an indirect sense. Your total metabolic rate is the rate of kilocalorie consumption to fuel for all ongoing activities. It increases with skeletal muscle activity and food ingestion. This is called food-induced thermogenesis, and it's greatest with protein and alcohol ingestion. How do we regulate our body temperature? Well, your body temperature reflects the balance between heat production and heat loss. At rest, the liver, heart, brain, and kidneys, as well as the endocrine organs, generate the most heat. During exercise, heat production from skeletal muscle increases dramatically. Normal body temperature is around 37 degrees plus or minus 5 degrees centigrade. Optimal enzyme activity is maintained at this temperature. Increased temperatures denature proteins and suppress neuron activity. Children less than 5 can have convulsions at 41 degrees centigrade, which is 106 Fahrenheit. 43 degrees centigrade or 109 degrees Fahrenheit will basically kill you. The tissues can tolerate low body temperatures to a point, and that point is that when chemical reactions slow to the point where they're no longer producing enough products for cells to use in order to perform life activities, the cell will shut down, the cell will die. So what you're looking at here is just a diagram showing you the activities that generate heat production and those that generate heat loss. And again, we want to have a homeostatic balance here. So let's talk about your core and your shell temperature. Your core are the organs within the skull, the thoracic and abdominal cavities, and it generally has the highest temperature. The rectal temperature is the best clinical indicator of your core. It's regulated and fairly constant. Your skin temperature, okay, it can fluctuate, okay. The blood is a major agent of heat exchange between your core and your shell. Your shell has the lowest temperature. It fluctuates between 20 to 40 degrees centigrade. As a result, what we see here is a method for regulating core and shell temperature, right? The blood can act as a conduit. As an example, okay, if we take a hot day and you were to go out into the pounding sunshine, what you would notice is that your skin would turn red and you would begin to sweat. The reason for this is that we have peripheral vasodilation. The sweat, as it evaporates, cools the skin. The skin, as it cools the blood. The blood, as a result, cools the body. And so it can circulate back to the core and drop the core temperature and maintain, again, that normal 98.6 that we need in order to survive. On a cold day, what happens is that we have peripheral vasoconstriction. The blood is redirected away from the periphery, away from the shell, and towards the core in an effort to attempt to keep your vital organs alive. And as a result, the skin becomes clammy. Sometimes we experience frostbite in extended stays in very cold temperature. But the body still accomplishes its major goal, which is to keep you alive. The four mechanisms of heat transfer include radiation, which is the loss of heat by infrared rays. This does not require a medium through which to propagate, so this can happen in a vacuum. Conduction, which is the transfer of heat by direct contact, and convection, which is the transfer of heat to the surrounding air. Evaporation is heat loss due to the evaporation of water from body surfaces. The heat that's absorbed by the water during evaporation is called the heat of vaporization. As the water transforms from a liquid to a gas, it takes with it kinetic energy. That kinetic energy, as it leaves the body, results in a heat flow out of the body and a cooling of the body's temperature. So here you can see an example of heat exchange. Here we have conduction and convection all at the same time. Insensible heat loss accompanies insensible water loss from the lungs, the oral mucosa, and the skin. This is about 10% of the basal heat production. Sensible heat loss is when the body temperature rises and sweating increases water vaporization. Your thermoregulatory centers are centered in the hypothalamus, in the preoptic region, which is the integrating center for thermoregulation. You have two thermoregulatory centers. You've got a heat loss and a heat promoting center in the hypothalamus. It receives afferent input from the peripheral thermoreceptors in the skin and the central thermoreceptors, which are in the core, some of which are actually within the hypothalamus. This initiates appropriate heat loss and heat promoting activity. How do we promote heat retention? Well, we can have constriction of cutaneous blood vessels. The sympathetic nervous system can stimulate this, and then shivering can produce heat through the hydrolysis of ATP. Increased metabolic rate comes as a result of epinephrine and norepinephrine secretion from the sympathetic nervous system and also from the adrenal medulla. This chemical produces non-shivering thermogenesis in infants. There's also brown adipose tissue in infants and adults. This is a very special type of fat that has an uncoupled electron transport chain, and so what happens here is that NADH and FADH2 donate high energy electrons to the electron transport chain that do not ultimately generate ATP. Instead, what they do through the movement of electrons down the chain in this uncoupled chain is they simply pump protons that leak back into the matrix, and the net result is to produce heat, but ultimately not ATP because the gradient never diffuses through the ATP synthase. Enhanced thyroxine release in infants can also promote an elevation in body temperature. There's also behavioral modification that can control body temperature. This includes putting on more clothes, drinking hot fluids, changing your posture, such as clasping your arms across your chest, and increasing physical activity, such as jumping up and down or running in place. Heat loss mechanisms are basically where the body begins to cool. The heat promoting center can become inhibited. This can result in the dilation of cutaneous blood vessels so the skin turns red. Enhanced filtration of the blood produces sweat. As it accumulates on the skin and evaporates, this cools the blood under the skin. Voluntary measures include reducing activity and seeking a cooler environment and wearing light-colored, loose-fitting clothing. And so what you're looking at here is just the mechanisms of body temperature. Okay, so what we're seeing here is the stimulus, okay, increased body temperature, the blood gets warmer, so we trigger the heat loss mechanisms. We have peripheral vasodilation, production of sweat, and as a result we cool the skin surface and cool the blood. The body temperature drops, and the temperature declines. The hypothalamus senses this, and it shuts off now these responses until what? Until you heat back up again. On the flip side, we've got a reduction in body temperature, right? Stimulus, decreased body temperature, signals the hypothalamus, which promotes now the peripheral vasoconstriction. The skeletal muscles begin to shiver, and then this generates heat. The body temperature rises, and now we're back where we were before until what? The body temperature drops again due to our presence in a cold environment. Hyperthermia is an elevated body temperature that depresses hypothalamic activity. There's a positive feedback mechanism which is meant to prevent heat stroke, and it begins at a core temperature of 41 degrees centigrade. This results in increased temperatures. The skin gets hot and dry, and the organs can become damaged at this point. This can be fatal if it's not corrected. In heat exhaustion, we have heat-associated collapse after vigorous exercise, and it can be due to dehydration and low blood pressure. You're losing water through perspiration, and as a result, your blood volume drops. The blood pressure can't be maintained. The heat loss mechanisms are still functioning, and it can progress to heat stroke if the individual is not cooled and rehydrated properly. And again, this rehydration should be in the form of an electrolyte-containing solution, such as a sports drink. In hypothermia, we have low body temperature from cold exposure. The vital signs begin to decrease. Shivering stops at a core temperature of between 30 and 32 degrees centigrade. Then it can progress to coma and death by cardiac arrest at around 21 degrees centigrade. Fever is controlled hyperthermia. It can be due to an infection or allergies or central nervous system malfunction. Macrophages release cytokines which have a temperature elevating activity. They're classed as pyrogens. This causes the release of prostaglandins from the hypothalamus. Prostaglandins reset the hypothalamic thermostat. Heat-producing mechanisms result in a rising body temperature. Natural body defenses or antibiotics can reverse this process. Cryogens, such as vasopressin, can reset the thermostat lower, and this leads to heat loss mechanisms, and this causes the temperature to begin to drop. So remember oxytocin and vasopressin, right? What does vasopressin do? Vasopressin has another name. Its name is antidiuretic hormone. And what does antidiuretic hormone do? It causes you now to retain more fluid, okay? Retain more fluid. The result now is to control this heat production. There's also inborn errors of metabolism that can result in homeostatic imbalance and inability to properly execute metabolic pathways. Cystic fibrosis is basically an error in a pump that controls the distribution of sodium in the extracellular environment. It can have effects on the urogenital system as well as the respiratory system. In the respiratory system, it results in the production of a thick, salty mucus, which promotes bacterial growth, which can lead to the formation of scar tissue in the lungs and a resultant destruction of the alveoli. And this reduces the ability of the respiratory system to exchange oxygen for carbon dioxide, and the result then is inefficient loading of oxygen into the blood and a suppression of metabolic rate. Phenylketonuria is an inability to metabolize the amino acid phenylalanine. The cells, as a result, generate a metabolite that serves as a neurotoxin in the blood that can lead to brain damage and retardation within a few months. This is why on the back of every can of diet soda that uses aspartame as a sweetener, you will see not for consumption by phenylketonurics. This is a recessive disorder, so you have to have 2 bad copies of the gene in order for this disease to show up. And the way to treat this condition is to avoid anything with phenylalanine in it. Remember that phenylalanine is an amino acid, so you have to be very careful about the type of protein that you consume. Okay, what are some of the developmental aspects of metabolic disorder? Well, lack of calories, proteins, and vitamins in utero and during the first 3 years can result in mental deficits and cognitive deficits. Insulin-dependent diabetes and genetic disorders can lead to metabolic problems in children, and non-insulin dependent diabetes can occur in middle and old age, especially in those individuals who are obese. This is what's called insulin insensitivity type diabetes. The metabolic rate declines throughout your lifespan, and the reason for this is for the survival of the species. You have to remember now that relatively recently, in terms of evolutionary time, man didn't live in urban, suburban, and rural environments. Instead, he was a hunter-gatherer, and the way that he procured food was to simply go out and capture whatever was nearby and consume that meal. Basically, bring back the food item to the cave, and then we would divvy up the food amongst the members of the tribe. Those individuals with the higher metabolic rate were those who were young and most able to hunt. As a result, they got most of the nutrients. Those individuals that had a lower metabolic rate, the aged and infirmed, those least able to hunt, got a proportionately smaller amount of the food, and the result was that the tribe itself could survive because most of the nutrients were going to the individuals in the tribe who were best able to procure food for the group. If this were reversed and the metabolic demands of that population rose with increasing age and decreasing ability to hunt and gather, it would result in the elimination of that group of individuals because you would be giving the bulk of the nutrients to those individuals who were least able to procure the nutrients in the first place. So we've been evolutionarily selected to store fat. Okay, other components. A lot of meds for age-related problems can influence nutrition. Diuretics can be used for heart failure and hypertension, but they can increase the risk of hypokalemia. That's basically reduced potassium levels. Some antibiotics can interfere with digestion and absorption and cause diarrhea. And mineral oil can decrease absorption of fat-soluble vitamins, which are important cofactors in enzymatic reaction. Excessive alcohol consumption can lead to malabsorption and vitamin and mineral deficiencies, deranged metabolism, and damage to the liver and the pancreas. Other components. Non-enzymatic binding of glucose to protein can increase with age, and this can cause lens clouding, general tissue stiffening, and loss of elasticity, leading to things such as cataracts and arteriosclerosis, nephrosclerosis, and a decline in muscle activity, just to name a few of the degenerative effects that occur as we age. Okay, that brings us to the end of this chapter, so make sure to review this material in your text. It will be covered on the exam. See everybody in class.